Right, last time when we talked about how viruses encounter hosts and enter them, we talked a little bit about the non-specific defenses that are present. Today we're going to get into that in more detail. In fact, today and next time we're going to be talking about the main defenses against virus infection. So today we're going to start with innate and intrinsic defenses. So last time when we talked about viruses getting in the various mucosal surfaces, the skin, the eye, etc., we discussed very briefly the physical and chemical defenses that are present at those surfaces. So for example, the fact that the outer layer of skin is dead is a defense. The low pH of the skin, the antimicrobial peptides that are present, tears in your eye that are cleansing away particles, etc. These are all what I call physical and chemical defenses, or you'll see it in the literature, they're called holistic defenses. They involve the entire host. And they eliminate most viruses encountering us. But if any viruses should overcome those holistic barriers, they encounter a series of increasingly more difficult defenses, which are illustrated here. The first is intrinsic, and then innate immunity, and finally acquired or adaptive. And we'll talk about intrinsic and innate today, but each is an increasingly high barrier to an infection. More viruses are eliminated at each time, and after innate immunity, if there are still viruses replicating, then adaptive immunity kicks in. So today we're going to talk about intrinsic and innate immunity, and next time we'll talk about acquired immunity. So intrinsic immunity, which is also called cell autonomous, is always present in the uninfected cell. It doesn't have to be turned on. All the players that are part of it are always present, and a single cell can undergo these intrinsic defenses. And some examples that we'll talk about in a little detail include apoptosis, autophagy, RNA silencing, and antiviral proteins. And there are many others, but I just want to give you a sense of how this works. So that's intrinsic. Then we have innate immunity, which we define as something you have to induce. All right? Infection induces an innate immune response, and that is different from intrinsic, because again, intrinsic you don't have to induce anything, or induction is minimal. So we're going to talk about those two today. And adaptive immunity is tailored to the pathogen. So we make antibodies and T cells that are pathogen specific, and this takes about 14 to 21 days. And so while that's happening, intrinsic and innate immune defenses usually can take care of the infection. And they do. They take care of most infections. As you'll see, they're very powerful. By the time a virus gets to adaptive immunity, it's replicating quite vigorously and it's causing illness. All right, so intrinsic and innate, you should understand the difference between the two of them. So let's talk about a few examples of intrinsic immunity. Again, this is present in an infected cell. So virus particles come into your respiratory tract, and you have one cell infected somewhere in your tract. Those particles have gotten beyond mucus and all the other non-specific holistic defenses. A cell can do something on its own, and that's intrinsic defense. Now, one of the defenses that's ancient is RNA interference. And what happens here is that an incoming virus, if this is an RNA virus, the viral genome is recognized by a system which involves processing the viral RNA, and the enzyme here is called dicer. Dicer chops up the RNA into 21 to 22 nucleotide pieces. It's then made, one of the strands is removed, so now you have a single-stranded RNA, or a short interfering RNA. And this, together with a number of other proteins, including argonaute, comprise the RISC complex. Now that RNA is complementary to the original genome of the virus that came in. It will hybridize to the viral RNA, and then the RISC complex will direct degradation of the viral RNA. All right? So that is RNA interference involving short interfering RNAs derived from the viral genome. This was discovered in plants. It's known to be a viral defense mechanism in plants and invertebrates. Whether or not it works in mammalian cells isn't clear yet. A few papers were published last year claiming for the first time that RNA interference is an antiviral response in mammalian cells, but there's a lot of controversy over those papers, and nothing has come out since to confirm them. So whether or not mammals have this as an intrinsic defense is not clear. They may not need it. They have so many other things, as you will see, whereas plants and invertebrates don't have a lot of other defenses, and that may be why they predominate in those. Anyway, we'll see what happens in terms of mammals, but certainly in plants and in insects, RNA interference is a major antiviral system. Of course, if viruses didn't counter RNA interference, they wouldn't be around. So there are countermeasures to this system in plant and insect viruses. We don't need to talk about them, but many of them involve blocking the RNAi system, for example, dicer or the RISC complex. So as you'll see, this is going to be a theme throughout today. At every step of intrinsic and innate immunity, viruses have evolved genes to counter it. Otherwise, they wouldn't be here. The viruses that don't have countermeasures left us many, many years ago. So let's move on to some systems that we know work in mammalian cells. This is an example of an antiviral protein that's present in the cell. Doesn't have to be induced. It happens to be specific for retroviruses. It's called APOBEC3. This is a member of a family of proteins. The word stands for apolipoprotein B mRNA editing catalytic polypeptide. And APOBEC3, which is abbreviated A3G, is a protein that gets incorporated into HIV virus particles, human immunodeficiency virus particles. These are retroviruses. Here is a particle at the bottom here with the two pieces of RNA, the two plus-stranded RNA genomes. It's an envelope virus. And APOBEC is the orange ovals that's gotten packaged in there. And what happens is when this virus infects another cell, remember the RNA genome is reverse transcribed to give you a double-stranded DNA, then APOBEC deaminates the DNA. All right, what does that mean? Here on the lower right is an explanation. Here's the viral RNA with some bases GG and GA, and these are going to be copied into minus strand DNA. It'll be CC and CT, but then the APOBEC deaminates the C, so it becomes a U. So it just removes an amine group from the C. Now it's a U, and so when it's copied to make the second strand, instead of having a G, you're going to have an A. So every C that is deaminated, and many of them are, gets changed, and therefore you mutate the virus out of existence. This has been called, this protein, APOBEC3G, has been called a WMD, a weapon of mass deamination. Okay? And now the thing is, how you say, does HIV actually replicate? Well, the virus has a protein called VIF, right there, the little blue protein. And VIF is present in the cell in which the HIV is being made. VIF makes APOBEC get ubiquitinated. And you may know that when you have ubiquitin moieties attached to a protein, it targets it for degradation by the proteasome. So APOBEC is degraded. So in the presence of VIF, the virions that are made don't have APOBEC in them, and they're fine. So the pathway I showed you on the bottom happens with a mutant virus lacking VIF. So that's what would happen. So the virus has evolved this gene VIF to counter it. And VIF is an important host range determinant. So for example, gorillas have been infected once, as far as we know, with a virus from chimpanzees, a simian immunodeficiency virus, only once. Whereas chimps have been infected a number of times. And the reason is that the gorilla APOBEC can't be antagonized by chimp SIV viruses. So this is a very important determinant of host specificity. Even though HIV gets around it, in other viruses it's important. So this hints to us a strategy for antiviral control. If we could somehow antagonize VIF, for example, that might be one way to get around this. All right, so that's one example of an intrinsic defense. So that protein is always present in the cell, perfect example of intrinsic. Here's another one, autophagy. We've mentioned autophagy before. This is a process by which if a cell undergoes stress, which can include virus infection, it responds by engulfing the cytoplasm in vesicles. So you start with this crescent-shaped double membrane vesicle. It engulfs the cytoplasm, and then these vesicles, called autophagosomes, fuse with lysosomes that digests the contents, and then eventually these vesicles containing degraded contents go to the plasma membrane and they release them. It's a kind of way of recycling contents, right? So when viruses are present in the cells, they get engulfed in these autophagosomes, either virus particles or viral genomes, and they are degraded. All right, so that is another intrinsic defense of the cell to a virus infection. It destroys it, and in so doing may survive. Another one is apoptosis, which is very different from autophagy because in apoptosis, the cell dies. It commits suicide. This is programmed cell death because it senses that something isn't right. It could be a virus infection or some kind of other stress. And there are a series of events that happen to trigger apoptosis, a very complicated pathway, in fact, a number of different pathways. Here's a normal cell, begins to go through apoptosis. The DNA becomes condensed. Eventually, pieces of the cytoplasm come off. These are called apoptotic bodies, and this cell will die. But these apoptotic bodies are picked up by what we call sentinel cells. These can be macrophages or dendritic cells, and these are important cells in the innate response, as we will see. So these cells constantly circulate in your body, and they're always looking for apoptotic bodies. And when they encounter them, because we undergo apoptosis all the time, even if we're not infected, if you want to get rid of cells, for example, they can be apoptosed. So these macrophages go around picking up apoptotic bodies, and they ask, is there something foreign in here? Do I need to worry about it? And the way they do that will become very clear in this talk in a few moments. They can sense if there are foreign molecules in these apoptotic bodies. So if this cell has been virus infected, virus infections often trigger apoptosis. These bodies will contain viral nucleic acids and proteins. They can be sensed by the macrophage or the dendritic cell. And then the macrophage of the dendritic cell will respond in ways that we'll talk about in a moment. These sentinel cells, which is what we call macrophages and dendritic cells, can also go to the lymph node. So wherever there's an infected area, there's a lymph node not too far away, and they will present the protein that they've picked up in these apoptotic bodies to the T cells in the lymph node, and they'll say, is this foreign? And if so, then the adaptive response will kick in. We'll cover more of that in a moment. So this is very important. Apoptosis is monitored by sentinel cells. If these cells are infected, they want to know what's in there and if they need to respond. We'll come back to this over and over. Again, apoptosis and autophagy are antagonized by viral gene products. And a list of the the viral regulators of apoptosis are shown here. And I don't want you to memorize them. I want you to be amazed that there's so many ways that viruses have evolved to antagonize apoptosis. So here are different viruses in the middle here. You can see they're mostly DNA viruses. They're mostly DNA viruses of various sized genomes. And here are the different cellular targets. So apoptosis is a complicated process that has many steps, and viral antagonists operate at different steps. So for example, caspases are important enzymes that modulate apoptosis, and viruses encode inactivators. And there are various receptors and ligands that are important for apoptosis. Viruses make antagonists of those as well. And in fact, one virus, cytomegalovirus, makes an RNA that inhibits apoptosis. It's a non-coding RNA, it doesn't encode a protein. Yet that RNA can bind a protein that is needed for apoptosis. I'm going to hammer on this over and over again. At every step of immune defenses, there's a viral countermeasure, and this is just one example. These are not even all the examples of how viruses can overcome apoptosis. Now, there's one step on here that I want you to note, because this will be important in a couple of lectures when we talk about transformation. The cell cycle is one target of inhibitors, and you can see here that P53 is mentioned 3 times as the target of an inhibitor of apoptosis. One blocks P53-mediated apoptosis, another one degrades P53, and another one inactivates it. So P53, you probably have heard of this. It's a very important cell cycle checkpoint protein. And it triggers apoptosis when it thinks that the cell isn't doing the right thing. Now, if you remember from DNA replication, remember the DNA viruses that replicate their genomes using the cellular apparatus, they kick the cell into mitosis, remember, because most of our cells are not dividing. So viruses have proteins like T antigen that make the cells go into mitosis. P53 monitors that. And P53 knows when the cell should be dividing. And if it senses unscheduled DNA synthesis, such as kicked on by a viral genome, it says, we're going to apoptose. That's it. This is not happening in my cell. So P53 induces apoptosis. And these viral antagonists are blocking that. Why it does so will become clear, as I said, in a couple of lectures, but I just want you to take a look at that. All right, apoptosis. Bacteria also have intrinsic defenses. This is a relatively recently discovered phenomenon called CRISPRs. And this is clustered, regularly interspaced, short palindromic repeats. And you may have heard that a couple of scientists just got the what prize? What's the name of that prize? $3 million each. Innovator Prize. It's founded by Mark Zuckerberg and Sergey Brin. And so they got it for discovering, Jennifer Doudna and Charpentier both got it for discovering CRISPRs. This is an intrinsic defense of bacteria against foreign DNA. So here we have a phage, a bacteriophage, a bacterial virus infecting a bacterium. All right, we're shifting to prokaryotes for a moment. This phage injects its DNA. Of course, the DNA is going to replicate and so forth, but the bacterium has a defense. It has a system whereby a series of proteins that have enzymatic activity will chop up some of that genome and integrate it into the genome, the DNA of the bacterium. And over time, if the bacterium survives, it has a history of all the foreign DNAs that have come in, plasmids, transposons, viruses. It keeps this very long array of repeated sequences called the CRISPR array. That's how they were first discovered. And each of them represents an insult from an original incoming DNA. So it's kind of like memory. And then when this virus comes back, this array is transcribed into an RNA, which is then processed by additional components of this CRISPR machinery. And you have these processed complementary RNAs. And these will hybridize to the incoming viral DNA and lead to its degradation. So again, they're enzyme associated with this that inactivate or chop up the DNA. So the original memory of infection is made into an RNA that guides the degradation. So kind of like RNA silencing, if you will. And this one has memory because these are integrated in the genome. This is basically an intrinsic response because it's present in the cell and it doesn't need any inducing. Now this has become, the CRISPR system has become incredibly powerful for use in experiments in the lab. You can inactivate genes very nicely with this. You can have a gene clipped out of a genome, for example, and it's being used for gene therapy in people. We're going to have a talk on gene therapy later on in this course, and we're going to talk about ways that you can do gene editing using these CRISPR sequences. So this is really, really a big deal. All right, this first question, intrinsic defenses are always present. Which of the following are included? Intrinsic. Antibodies, T cells, autophagy, all of the above, or none of the above. So 82% of you picked C, which is autophagy, and that is correct. That's an intrinsic defense we just talked about. Antibodies and T cells are not intrinsic defenses. No one picked those individually, but a few of you picked all. and some of you pick none, and autophagy is the one. So the next defense system is innate immunity. Perhaps more of you are familiar with this. Certainly if you took the immunology course, you should know about this, but we're going to look at it from a virus perspective. This is something that happens very quickly after infection. So even if we have intrinsic defenses, innate immunity is kicking in as well. So it can be activated very shortly after infection. And it comprises cytokines, which are proteins produced by various cells and which bind to receptors on cells to have their effect, and various sentinel cells, that includes dendritic cells, macrophages, which we talked about a little bit, NK. NK stands for natural killer cells. These are all part of the innate immune system, and complement, a collection of serum proteins that actually bridge innate and adaptive immunity. So innate immunity has its own ways of overcoming virus infection we're going to talk about, but it also collaborates with the adaptive system. When it gets overwhelmed, if you will, it can inform the adaptive system and say, we need some help here, we can't do this on our own, we can't handle this infection. And it will tell the adaptive system to make antibodies and T cells. So let's see how this works. So let's say we have our original infected cell in our respiratory tract. One cell maybe, or a few, got infected with a virus. Intrinsic defenses are acting, but maybe they can't stop the infection. This infected cell can produce some cytokines on its own. It's sensing, depending on how quickly the infection proceeds, it can produce some cytokines, and that can attract components of innate immunity. And a key player in this cell knowing it's infected are what we call pattern recognition receptors. And these recognize the pathogen as foreign. And part of the way this is done is in the macrophage or dendritic cell, but can also happen in the original infected cell. We're going to talk about these in a moment. The pattern recognition receptors, if they say, oh, wow, this is not foreign, that's the end of it, there's no innate response. But if they recognize that this cell has been infected with if something foreign, it produces viral RNA or viral protein, the innate response will be activated. And again, this includes the production of cytokines, and we'll talk about the roles of dendritic cells and NK cells and complement in the innate response. And sometimes that clears the infection. But if it doesn't, then the innate response informs the adaptive response, which leads to production of B and T cells. And this adaptive response we'll talk about in detail next time. So the recognition of a foreign virus infecting this original cell can happen within that cell. That cell can sense it. That cell could apoptose and release apoptotic bodies, which would then be sensed by macrophages or dendritic cells. Right? And those could be brought to the infected cell because the infected cell will produce cytokines, and many of those tell cells to come there. So it's a very wide-ranging response. And the key is it's very quickly can go from one cell to involving the entire organism in defense. So let's talk first about how the innate system recognizes something that's not self. This is a key difference between the innate system and intrinsic immunity. This begins in 1980. Two scientists in Germany, Nusslein-Volhard and Wieschaus, they were studying Drosophila, the fruit fly, to figure out how it developed. They were trying to identify genes that played roles in development. And they were doing this by making fly mutants and seeing how they develop. And they've identified a gene that established the dorsal ventral axis, a very important developmental stage in the fly. And this was called the Toll gene, because legend has it that they were looking at these flies in a microscope, and these were German investigators, and they looked at them and said Toll, which means crazy far out in German. And so that became the name of the gene. You know, in Drosophila, you can call genes interesting things. And they got the Nobel Prize for that 15 years later. And in 1996, the Toll gene, besides having a role in development of the fly, was shown to have a role in immunity to fungal infections in the fly. So this got people thinking, I wonder if this Toll gene is in mammals and whether it could also recognize foreign invaders. And it turned out that in 1997, Toll-like receptors were identified in mammals. This whole field was driven by a guy at Yale named Charles Janeway. who had an idea years before this protein was discovered. There have to be pattern recognition receptors, he said. There have to be some kind of receptor in a cell that will recognize a foreign virus or bacterium or fungus. And when these were discovered, he said, that's it. So these are the so-called toll-like receptors. And now there are a whole number of them, as you can see here, 1 through 11. And they recognize different things like triacyllipoproteins, double-stranded RNA, lipopolysaccharide from bacteria, flagellin from bacterium, single-stranded RNA, and DNA as well. So they can recognize different things as being pathogen-specific. So that's why we call them pattern recognition receptors or pathogen recognition receptors, PRRs. And these are transmembrane proteins. So here's the original toll protein from Drosophila, also called Spätzle. And it's made up of a series of leucine-rich repeats. So each of those horizontal boxes is a sequence of protein which is rich in leucines. And it's repeated over and over. And you can see in all these other toll-like receptors, the leucine-rich repeat is present as well. And this is a transmembrane protein. Then there's a cytoplasmic domain called the TIR domain, which stands for toll IL-1 receptor domain. So interleukin-1 is a cytokine that binds to a receptor and signals through a cellular cytoplasmic domain that looks a lot like the toll-like receptor domain. All right, so all of these are individual toll-like receptors that recognize different molecules as foreign. Here's actually the crystal structure of the extracellular domain of this toll-like receptor. So each of these individual repeats is the leucine-rich repeat, forms a curved structure. And the ligands typically bind within this area. You can see a couple of ligands binding in here. And this is how the cell detects something as foreign, by these toll-like receptors. So here's an example of how this works. Here is a cell where we have an endosome. Many of these toll-like receptors are produced in endosomes. These are the ones that typically detect virus particles. Other TLRs are on the plasma membrane, but we don't think they're as involved in detecting viruses. So here is an endosome with 3 different TLRs, 3, 7, 8, and 9. And they're detecting various viral signatures like double-stranded RNA, single-stranded RNA, DNA. And remember, the endosome is where viruses enter, many viruses enter the cell. So this is a good place to put sensors. So the entry process is a little bit leaky. Some nucleic acids get out into the endosome, and those will be detected by the toll-like receptors. So that's part of the normal virus entry path, right? Viruses bind receptors. They're taken up into endosomes, and in the endosomes, they can be sensed by toll-like receptors. There's also another way that bits of viruses get into the endosome. If this were a macrophage or a dendritic cell, remember, it would be sampling us for apoptotic bodies. These are pieces of virus-infected cells that would contain virus protein and virus RNA. Those are taken up into the macrophages and dendritic cells by endocytosis. And here they are coming in to this cell by endocytosis. And also autophagy within this cell could give rise to material getting into the endosome. So when toll-like receptors recognize a nucleic acid or a protein as foreign, they respond by inducing the production of cytokines. And this goes through a series of signaling pathways. So the cytoplasmic portion of each of these toll-like receptors is bound to an adapter protein here. And these initiate a series of phosphorylation cascades, which ultimately result in transcription proteins going into the nucleus and causing transcription of mRNAs for cytokines. So here, for example, the end result in this particular pathway is phosphorylation of IRF7, which is a transcription protein. The phosphorylation makes it go in the nucleus, where it then turns on the synthesis of cytokines called interferons. And you can see there are other pathways involving the NF-kappa B pathway. Here we have NF-kappa B being separated from its inhibitory subunit, goes in the nucleus, turns on the synthesis of cytokines, and a separate pathway involving IRF3 as well. So you have TLRs in an endosome. They sense viral nucleic acids. They initiate a signaling pathway. You get transcription of mRNAs encoding cytokines. The cytokines have a variety of roles. They will they'll be translated, the mRNAs will be translated into cytokine proteins. They'll be secreted. Some of those will attract cells to the infected area. They will bring in macrophages and dendritic cells and other NK cells. Some of the cytokines will turn on other genes that have antiviral properties. We'll talk about this in a moment. All right, so that's how toll-like receptors work. There are other systems in cells for sensing viral patterns. So not everything goes through an endosome, right? Some viruses put their nucleic acid in the cytoplasm. So there are cytoplasmic proteins we call cytoplasmic helicases because they were first found to unwind RNA. There are two of them, RIG-I and MDA5. So they're in the cytoplasm. They're proteins, and they detect RNA. RIG-I detects RNA with 5' phosphates. Cellular RNAs do not have 5' phosphates. So this tells the cell that if there is an RNA with a 5' phosphate, it's viral. It's viral, and then RIG-I is going to act, again, through a signaling pathway to turn on the production of cytokine mRNAs. MDA5 is another cytoplasmic helicase. It detects double-stranded RNA. Again, double-stranded RNA isn't found in an uninfected cell. This is a virus product. MDA sees double-stranded RNA. A virus must be present. It's activated. Both of these proteins are activated by the presence of their ligands. Activation allows them to interact with a mitochondrial protein called MAVs, and then they go down different pathways of activation, of transcription, depending on the particular cell type and the virus involved. And again, the end result is the production of cytokine mRNA and interferon mRNAs. Interferons are a kind of cytokine that we'll talk about in a moment. So again, sensing in the endosome or in the cytoplasm gives rise to the production of cytokines. This is the primary defense of the innate immune system. So this can happen in your original infected cell in your respiratory tract. It can also happen in dendritic cells or macrophages that pick up apoptotic bodies and sense that there is foreign material in them. They can produce cytokines as well. You can also sense DNA. This is a more recent discovery. There is a protein in the cytoplasm of the cell called cGAS. This is short for cyclic GMP-AMP synthase. So this is a cytoplasmic protein that will detect viral DNA in the cytoplasm. It probably collaborates with a couple of other cellular proteins, which are shown here. And when it detects viral DNA, it's not clear how it knows that it's viral and cellular yet. Okay, the presence in the cytoplasm has been argued by some to be the differentiator, but I'm not sure that that's correct. But let's assume that cGAS somehow knows that it's viral. It then takes ATP and GTP and synthesizes this very unusual cyclic molecule called cyclic GMP-AMP. And that's shown in the upper right here. So it's an A and a G joined by this cyclic phosphate ring right in the middle there, cyclic GAMP. Yeah, and that can be viewed as somewhat of a second messenger. It collaborates with another cellular protein called STING, and that turns on the same kinds of signaling pathways that we've just looked at for the TLRs and the cytoplasmic helicases, and turns on cytokine synthesis in the nucleus. So it's a very unusual molecule, a small molecule produced when DNA is sensed. You can almost look at it as acting the way hormones do. On the right-hand side here is how a hormone would bind to a receptor and induce the synthesis of cyclic AMP, right, by adenylate cyclase. And cyclic AMP then goes through downstream messengers to induce a physiological response. So here, the detection of DNA results in the production of this small molecule, which also has downstream effects as well. It's really very interesting. So you can detect many different molecules by these various procedures. Now, as you might guess, viruses have evolved to counter every one of these steps that I have told you about and more. And this is a summary of all the antagonists of helicases and TLRs. And the red and the blue lettering, these are individual viral proteins that antagonize at every step. And again, you don't need to know each one of these. That would be crazy. But I want you to be amazed. There's over 100 different antagonists on this slide, starting from Let's see, here's MDA5 and RIG-I, which sense RNA, right? They are antagonists of RNA. They bind RNA so it won't be sensed. They are antagonists of each of these proteins. Some of these antagonists cleave the sensors, some of them just bind to it and block it. That mitochondrial protein, that is the beginning of the signaling pathway through RIG-I and MDA5, they're antagonists of that as well. And that whole signaling pathway, at every step of the way where there are phosphorylation of different intermediates, look, there are viral proteins that interfere. There are even viral proteins that interfere with transcription in the nucleus of the cytokine genes. And on the right is the TLR story. Again, antagonism at every step of the way. So again, this tells us that this innate immune defense is incredibly powerful, because if the virus doesn't antagonize it, it's not going to be around. All right, which of the following allow the innate immune system to distinguish microbes from self? Cytoplasmic helicases and toll-like receptors, antibodies, apoptosis, APOBEC, all of the above. Okay, 83% of you picked A, which is the right answer. Cytoplasmic helicases and TLRs. Remember, the question is, which allow the innate immune system to distinguish microbes from self. Some of you said APOBEC, but that's an intrinsic defense. That is present in the cell. It doesn't have to be induced as the components of innate immunity do. And antibodies certainly do not enable the innate immune system to distinguish microbes, because antibodies are produced later in infection. Now, one of the cytokines produced by sensing that we've talked about is interferon. And interferon is a collection of proteins originally discovered in 1957, when these investigators found that you could put inactivated influenza virus onto cells, chicken cells, and the supernatant of the cells had a factor in it that blocked infection of another cell. And they called this interferon for obvious reasons. We now know that this is made by virus-infected cells and also uninfected cells in response to the products that are detected in cells. So TLRs and cytoplasmic helicases, when they find a foreign molecule, they make cytokines, and one of those cytokines is interferon. So here at the bottom is a picture of this. We have an epithelial sheet. Respiratory, for example, virus is getting in and replicating. The cells detect the virus as foreign. They're producing cytokines. Among those are interferons, which can attempt to inhibit replication, as we'll see in a moment. Cytokines will also pull in dendritic cells into this area, macrophages. The macrophages can make more interferons. The dendritic cells can make more interferons in an attempt to stop infection. There are four main types of interferon. There are actually others as well we're not going to talk about. There's interferon alpha and beta, which is made by most cells. So that epithelial cell sheet infected in the previous slide is making interferon alpha and beta. It's induced by virus infection, again, by the sensors. There's interferon gamma, which is mainly produced by immune cells like T cells and NK cells. In fact, gamma is used as a marker for T cell activation. There's also interferon lambda, a more recently discovered type, mainly produced by epithelial cells. So this may be another main defense at epithelial barriers, the interferon lambda, produced by virus infection. So how does interferon work? So remember, the cell is infected. It produces cytokines as it detects a foreign presence. Among those cytokines are interferons. These are proteins that get secreted from the cell. So they're shown here as red squares and yellow octagons. So they're now outside of the cell. There are receptors on cell surfaces for the different kinds of interferon. And those interferons bind to the cell receptors and instigate a signaling pathway that leads to the production of new mRNAs. So this is a very rapid response. Within hours of infection, interferons are produced. They bind to receptors. And that binding leads to the synthesis of over 1,000 different cell proteins. These are called ISGs, or interferon-stimulated genes. So that's how this works. The interferon, for example, type 1, binds to its receptor. That binding leads to phosphorylation of the STAT proteins, which then go in the nucleus and assemble at the promoter of these interferon-stimulated genes. Same thing for type 2 interferon, which binds to a different receptor. these ISGs, so again, there are over 1,000. They're turned on by interferon. So interferon itself doesn't inhibit any virus by itself. All its job is to bind interferon receptors and turn on the synthesis of all these ISGs. It's the ISGs that actually have the antiviral activity. It's a hot area of virology right now to figure out what ISG inhibits your particular virus and how does it work, because we know very little about these. I just want to go through a couple of these ISGs to give you an idea of how they work. So here's an infected cell where a virus is getting in and replicating and budding. And in red are where some of the ISGs work that we know about. For example, there are two ISGs called OAS and RNA cell. We'll talk about those in a moment. These degrade RNA. And they can work in the cytoplasm or in the nucleus. MX is another protein that inhibits multiple steps in virus replication. It inhibits RNA synthesis, either cytoplasmic or nuclear. PKR. Does anybody remember PKR? You forgot it already, right? From a previous lecture. So PKR is an ISG. It's induced by interferon. It phosphorylates EIF2 and shuts off translation. So the cell is trying to turn everything off so it would stop virus replication. So PKR interferes with translation. Another interesting one is called ISG15, a small ubiquitin-like protein that's conjugated to viral proteins, probably helps degrade them. Viperin blocks virus budding, and Tetherin blocks the release of viruses who are about to bud and go off of the cell. These are just some of the 1,000 or so ISGs that we understand. Here are a couple in some detail. Interferon induces two proteins that work together, RNase L and 2' 5' oligo A synthetase. So what happens is this oligo A synthetase, it's induced by interferon. So a virus is infecting the cell. One of the many ISGs produced is OAS. It's inactive though. It needs viral double-stranded RNA to activate it, very much like PKR needs double-stranded RNA to activate it. When it's active, it then takes ATP and makes short stretches of oligo A from it. That's why we call it oligo A synthetase. That oligo A then activates RNase L, which is another ISG induced by interferon, which is inactive. The oligo A activates it, and now it can cleave RNA. It will cleave viral RNA, of course, which prevents from being translated. It also cleaves cellular RNA. So like many of these ISGs, it doesn't distinguish. The sensing distinguishes between virus and self, but the effectors of the innate response often do not distinguish. So the original infected cells may die, but that could spare the rest of the organism. So that's how these two proteins work together. Tetherin is another interesting ISG. Tetherin grabs viruses as they're budding from the plasma membrane. So here on the left of this image is a budding virus. And as it's about to be released, two Tetherin dimers. So Tetherin is a plasma membrane protein. It's inserted into the plasma membrane, and it grabs on to the virus membrane. So it has domains that sit into both the plasma membrane and the virus membrane, so the virus cannot be released. And it's then redirected to endocytosis pathways, and it's degraded. So this is a good way to prevent virus infection. However, viruses encode antagonists. If I don't remember to tell you that at every slide, it's true. And there are three shown here. There's an HIV protein, VPU, there's an Ebola virus, glycoprotein, and an HIV-2 protein. So these all antagonize Tetherin. They block its activity and allow the virus to bud from the cell. Another example of an interferon-induced protein is nitric oxide synthase. This is an enzyme shown at the top, NOS, nitric oxide synthase, takes arginine and makes nitric oxide from it, NO. Nitric oxide is highly reactive. When it gets near a protein, it will conjugate to it and the protein becomes inactive. So you can see at the top there, nitric oxide is being conjugated to this protein. The protein is drawn like a cloud. My pointer died, so I can't show you anymore. The cloud is the protein, and it's conjugated to nitric oxide. and now it's inactive. So this is made by sentinel cells. So on the bottom is an example. You have an epithelial sheet which is infected with viruses. Those are the red guys being produced. Remember, those cells are infected. They're making cytokines. The cytokines call out to macrophages and dendritic cells, come, help me out. So the macrophage is coming through the blood vessel at the top. It gets near that sheet, and it starts producing nitric oxide because nitric oxide synthase is induced by interferon. And that ends up killing a lot of the cells, but it localizes virus infection. Right, so that's another ISG. And basically, there are many more. We don't know how many of them work, but I think you get a clue that these are dangerous proteins. They kill cells, right? They destroy mRNAs. They lyse cells through nitric oxide. They are dangerous. So interferon is dangerous. It's a great system. It prevents a lot of virus infection. But all of our cells have receptors, so there's a lot of damage. And even if you have a respiratory infection, the interferon produced in your respiratory tract gets into your bloodstream and goes everywhere. And that's why you feel lousy when you have a cold or influenza. You have systemic symptoms like fever, chills, nausea, malaise. We call these flu-like symptoms, but in fact they are a consequence of most virus infections because most virus infections induce interferon. And so every infection gives you interferon production. Therefore, flu-like symptoms aren't just restricted to flu. So the next time you get a viral infection and you feel pretty bad, I want you to think about interferon because that's what's doing it. I want you to know what's going on inside of you. I think that's really important to do that. The interferon has spread from the original site, whether you have a respiratory infection or a GI infection. It's getting systemic, and you can't really prevent it because these are soluble proteins, right? There would be no easy way to limit the infection. Are there modulators of interferon? You bet. Here's a whole slide full of them. Again, the blue and the red are virus proteins that interfere at every step. So we have on the left, we have interferon with interferon signaling. So at the top left, we have interferon protein in blue binding interferon receptors. And then we have the signaling pathway leading to transcription of cytokine genes. There are viruses that make interferon mimics. These are proteins that look just like interferon. They bind the interferon receptor, but signaling doesn't occur. There are viruses that make interferon receptors. They're soluble and they bind interferon. And every step of the signaling pathway, there's an antagonist as well, and there are antagonists of transcription in the nucleus. Same with the effectors, the ISGs. Many of these ISGs have antagonists. You may remember PKR in the middle. There's PKR in the middle at the top. That phosphorylates EIF2 alpha and shuts down translation. I already told you about all the viral antagonists of that process as well. But they are antagonists of many other ISGs as well, shown on the right. And this is really incomplete because we don't even know how most of the ISGs work. So again, a testament to how powerful this innate system is, in particular interferon production, that you have so many levels of antagonism. Next question is, how do interferons limit virus replication? Interferons directly inhibit virus replication. They induce toll-like receptors. They induce ISGs. They damage cells. None of the above. So 93% said that interferons induce ISGs, and that's correct. The real key here is ISGs. I want you to understand that the way interferons work is by inducing these interferon-stimulated genes that then have antiviral effects. In fact, even the damage of interferon to cells isn't because of the interferon, it's because of the ISGs. Let's talk a little bit about sentinel cells. We've mentioned these already. These are part of the innate system. They include dendritic cells. There's a picture of dendritic cells on the lower left. These are recognizable by their long processes. These give them more surface area so they can sense more foreign protein. Macrophages, which we've talked about, which are highly phagocytic cells, and natural killer cells. On the right is a natural killer cell attacking another cell. The NK cell is in red. These cells are always moving around us looking for trouble, or signs of change to be more accurate. So they patrol you constantly. As you're sitting there listening to me, macrophages and dendritic cells and NK cells are slithering through your vessels and your tissues looking for trouble. And you don't even feel this, of course, but they're working really hard for you. So let's see how they work. So dendritic cells are, again, those very interesting looking, they almost look like neurons, right? They patrol various parts of your body. The intestinal areas, for example, the subepithelial tissues, the dendritic cells sit there. Sometimes they stick their processes through the epithelial cells into the intestine. There are dendritic cells patrolling various epithelia as well. They're in your body cavities. They're everywhere. They're moving around and constantly looking for a sign that you're infected. And that sign could be cytokines, or it could be packets of apoptotic bodies that have foreign proteins, because they can detect them. There are all kinds of dendritic cells. When they do pick up foreign proteins, I've already told you that macrophages and dendritic cells patrol our bodies looking for foreign proteins. They bring them to the lymph node to figure out if, in fact, they are foreign. So here you see dendritic cells coming into a lymph node, which is shown at the bottom. Wherever there's a virus infection, there isn't a lymph node very far away. And so the dendritic cells go to them. They know where to go. And in the lymph node, they ask the T cells if what I have here is foreign. So they will have, say, phagocytosed an apoptotic body. They will process the proteins in it and present it on the surface of the dendritic cell and ask the T cell if it's foreign. And then on the lower right is a picture of that happening. That's a picture in a lymph node. The dendritic cell is green. It's labeled with green fluorescent protein. The T cell is blue, and the red is blood vessels, capillaries. And the dendritic cell gets right next to it and presents the protein and asks, is this foreign? And if it is, then we have an adaptive response, which we're going to talk about next time. So dendritic cells are present in unactivated states, which are shown on the left here. They are pretty conventional looking cells, but they have lots of tools to work with the innate response. They have toll-like receptors. They have endosomes. They have cytokine receptors, and they can produce interferon. So if a dendritic cell picks up a piece of a virus protein, it can endocytose it, and the toll-like receptors can interrogate it and ask if it's foreign. The DCs can then make a ton of interferon right at the infection site to try and quell things. They can pick up cytokines from the infected cell, and these cytokines are a signature of virus infection, so the DC will make more cytokines in response. It can pick up apoptotic bodies and ask if there are foreign antigens in them. And when they do pick up an apoptotic body in an endosome, it will end up, the protein will be degraded, and a peptide derived from it will be loaded into MHC class II molecules. That's the little orange blob in those class II molecules. The process of detecting foreign signatures, foreign molecules, activates the dendritic cell, so it becomes a dendrite-like, which is shown on the right. It's now displaying the viral peptides on the surface in MHC class II molecules, and then it will go into the lymph node and present these peptides to the T cell. You have one here, a naive T cell being presented with this peptide, and then if it's foreign, then the T cells will become activated and become various helpers or effector cells. So these DCs are versatile. They can try to inhibit replication at the site of infection, and then they can also go into the lymph node to get the adaptive response started. And again, they can pick up pieces of viruses, they can pick up apoptotic bodies from the infected cell, they can pick up cytokines that tell them that there's a virus infection. So those are dendritic cells. NK cells are also part of the innate response, natural killer cells. They're the purple ones on the left here. They sense whether a cell is infected by two receptors on the cell surface. So you see the NK cell has what's called an inhibitory receptor, which is in red, and an activating receptor, which is in green. The ligand for the inhibitory receptor is MHC1 molecule on the target cell surface. And then there's a ligand for the activating receptor. So the top cell in the middle, the target cell, is uninfected, so neither MHC or the activating receptor are affected, so there's no cell lysis. That cell is detected as self and it's not infected. If a virus infects the target cell, one of the things that many virus infections does is to downregulate MHC molecules on the cell surface. So that second infected target cell now doesn't have any MHC1, and the NK cell detects the absence of MHC. It picks up the activating ligand, which sends a signal into the cell to the NK cell. The NK cell releases granules which induce apoptosis in that infected cell. Many virus infections also upregulate other cell surface proteins, and that's shown in the bottom, so they can upregulate, which are sensed as activating receptors by the NK cell. They will cause granule release and apoptosis. So this is how NK cells recognize an infected cell. They don't actually see antigen on the surface of the cell, as T cells do. We'll see how that works next time. These do not recognize viral antigen. They recognize an infected cell by the absence or upregulation of these specific cell surface proteins. So again, these are early workers in the innate response. These contribute to attempting to eliminate infection. Yes, viruses modulate NK cell activity at every of the steps that I've told you. For example, many viruses make MHC homologs. Those are shown all the way on the top. So on the left is a virus-infected cell. On the right is the NK cell. And so remember, there are receptors on the NK cell for the MHC molecules. Viruses will make MHC mimics so that the NK cell thinks that there's an MHC molecule there. The NK cell has an activating receptor. That's the third one down on the right. And remember, that has to interact with a ligand on the infected cell. Some viruses will make ligands that mimic the presence of this ligand on the cell surface, so the NK cell doesn't lyse it. So it goes on and on. There are, at every step of the NK interaction with the target cell, the virus can interfere. Because for some viruses, NK cell lysis is very important in their control. The final part of the innate response that I want to talk about is complement. This is a collection of serum proteins that were originally discovered a long time ago because they help antibodies lyse bacteria. It turns out they also function in virus replication. And there are many, many, many different proteins in the complement pathway. But the key point is that some of them can recognize virus proteins as foreign and initiate the production of cytokines. So on the upper left, for example, C1q is a pattern recognition protein, much like TLRs and the cytoplasmic helicases. C1q can recognize virus particles either by free particles in the cell fluids, and it can bind to them and help them be taken up by macrophages in a process called opsonization. The C1 particles can also recognize viral antigens on the cell surface, so infected cells display viral glycoproteins, and C1q can recognize those. Can also bind to antibodies that are bound to viral antigens on the cell surface. That's shown diagrammatically on the left there. All of this results in a multimerization of C1q and the initiation of a cascade of activities that involve processing of proteins, the liberation of what are called here mediators of inflammation. These are simply cytokines. So the complement pathway can generate cytokines, which will bring in other cells. And eventually, they can result in the formation of that complex on the right called a membrane attack complex. And this will actually poke a hole in the infected cell, destroying it. Okay, so complement detects a virus infection. This is only one of the 3 detector systems and can result in production of cytokines, which you've already talked a little bit about in their importance, and can poke holes in the cell. And yes, there are viral modulators of the complement cascade. Almost all of these complement proteins has a modulator that antagonizes it. Complement has 4 functions. It can lyse cells by the formation of that membrane attack complex. Can produce cytokines, and this leads to what we call inflammation, which we'll talk about in a moment. The C1q can coat virus particles so that they can be better taken up and destroyed by macrophages. And later on, when the adaptive response has kicked in and you're making a lot of antibodies, sometimes these kind of bind virus particles and get stuck in tissues because they make big complexes. Complement can help keep that soluble. So they solubilize antibody-antigen complexes so tissues are not compromised. The next question is, what is the main role of dendritic cells in virus defense? The dendritic cells destroy virus particles. Dendritic cells sense infected cells and produce interferons. Dendritic cells only instruct the adaptive response. Dendritic cells lyse virus infected cells, all of the above. The main role of DCs in viral defense. So 48% of you said DCs sense infected cells, I'm sorry, 50%, 57. Most of you said B, DCs sense infected cells and produce interferon, and that's what I'm looking for, their main role in viral defense. Now, some of you said they only instruct the adaptive response. They don't only instruct the adaptive response, absolutely not. That's why I put only in there, because they do make interferons which can resolve the infection on its own. All right, so let's end up by talking about cytokine production and what we call inflammation. Now, inflammation is a consequence of infected cells producing cytokines. I have my pointer for a few moments. So when cytokines are produced, we say the consequence is inflammation. And inflammation is composed of 4 signs: redness, pain, heat, and swelling. And this was recognized by the Romans a long time ago. And the Roman encyclopedia Celsus called them rubor, dolor, calor, and tumor. Redness, pain, heat, and swelling. And this is what cytokines do. You get increased blood flow to the infected area. The capillaries become permeable to let in immune cells in and out so they can clear the virus infection. You have cells coming in, phagocytic cells, eventually white blood cells. This makes the area swell. And you have tissue damage. So that gives you the redness, the pain, the heat, and the swelling. So this is all triggered by cytokines, not just interferon, but all the other proteins that we haven't talked about, the interleukins, the chemokines that are released at the site of infection that recruit immune cells to the area that have all these effects on permeability to try and clear the infection. So again, if you have, this is very common for skin infections. If you have a bacterial skin infection, you have a boil, you get all these signs all at once, and you absolutely know it. But this can happen in a more generalized manner, but it's all inflammation. So cytokines produce inflammation by having these effects. There are 3 different kinds of cytokines. There are what we call pro-inflammatory, so they would promote those 4 signs of infection. They include IL-1, tumor necrosis factor, IL-6 and 12, those are just some of them. And what these do is they activate white blood cells that are being brought into the area. Some of these cytokines are chemotactic. The white blood cells come in, leukocytes, then they start trying to clear infection. And that is pro-inflammatory because that is accompanied by those 4 signs of infection. Now, once the infection is over, you have to turn everything back down again. So we have anti-inflammatory cytokines like IL-10, 4, TGF-beta, and these suppress the pro-inflammatory cytokines. PIC stands for pro-inflammatory cytokines. So these antagonize the ones that promote inflammation. So they come up towards the end of an infection. And finally, we have chemokines like IL-8. These recruit cells to the infected area. So again, they're produced by an infected cell at an epithelial sheet, say, and they are recruiting dendritic cells, macrophages, leukocytes to the infected area to try and help clear the infection. And a key point here is that these cytokines, with some exceptions, are typically produced locally in the respiratory tract, in the intestinal tract, in your eye, but they're diffusible. They are soluble proteins that get into the circulation and they spread and have global effects in the so-called flu-like symptoms and other signs like sleepiness, lethargy, muscle pain, no appetite, nausea. These are all caused by the cytokines. They diffuse throughout. So for example, here we're showing a variety of pro-inflammatory cytokines being produced. They'll get into your brain. There's no stopping them, and they'll make you sleepy. They cause fever. They make you tired. They will also go elsewhere and do good things. They will get into your bone marrow and stimulate hematopoiesis, the production of blood cells like lymphocytes, which you need to combat the infection. So that makes sense. They will get into the liver, and they induce the synthesis of what we call acute phase proteins. These are other innate proteins that have antiviral effects. So there's a necessity for these cytokines to spread globally, but unfortunately the side effects is that you feel crappy. But when you get these effects, it means that the innate system is responding, so it's a good thing. These cytokines all work by binding to receptors, whether they're pro-inflammatory, anti-inflammatory, chemotactic, they bind to a receptor. Here's just one example, tumor necrosis factor, which is a major pro-inflammatory cytokine. There's a receptor on the cell surface called the TNF receptor, and depending on the particular cell type and the environment, binding of TNF can lead to apoptosis. So think of it, infected cells are producing TNF, it's going to diffuse and apoptose the neighboring cells, perhaps to prevent the virus from spreading. It can result in necrosis, another form of programmed cell death, or it can result in the induction of the transcription of cytokine mRNAs in the nucleus, depending on what is needed. So all of these have similar operations. They bind receptors. You have signaling pathways that lead to the final effect. So that's just one example, TNF. Every cytokine has its own receptor on the host cell. And so you can imagine that the particular effect of a virus infection really is going to depend on what kinds of cells are producing what cytokines and what receptors are involved. And yes, there are cytokine countermeasures encoded by virus genomes. Viruses can interfere with cytokine and chemokine synthesis. They can interfere with transcription of the genes, with production of them. Some of them need processing by proteases. Viruses can interfere with that. They can interfere with the action of cytokines. Some viruses include homologs. I've told you about the interferon homologs that bind interferon receptors, and they don't signal, so you don't get ISGs produced. But many viruses produce other cytokine homologs, like an IL-6 homolog and IL-8. So these are soluble mimics of the cytokines, but when they bind the receptor, they don't have the same biological effect. Of course, the virus wants to do that. It wants to block a pro-inflammatory response. Some viruses also make soluble cytokine receptors. So these are much like the receptors in the membrane for cytokines, like the TNF receptor I just showed you, except they're soluble. So the effect is they sop up all the cytokines and they never get to bind to their receptor on the cell surface. So again, just like every other step, amazing countermeasures that viruses need, otherwise they're eliminated. So let's summarize this in a tissue very briefly. So here we have an individual who has an infection, and in the middle of this picture, between the lymph vessel and the blood vessel on the bottom, there's an area called antigen. That's virus-infected area. These are cells that are producing virus particles, viral RNAs, viral proteins. They're sensed by the initial infected cells. Those cells produce cytokines. They bring in dendritic cells to the area. They bring in phagocytes, mast cells, basophils. Each of those produces its own complement of cytokines. Interferons eventually get involved in the middle there. NK cells are brought in by chemokines. Then some of these cells make proteins that make the capillaries permeable, like the eicosanoids produced by the phagocytes. You have cells coming in from the circulation, phagocytic cells, neutrophils, to try and clear the infection. And then you have cytokines produced like IL-1, TNF-alpha, interferon gamma, shown at the bottom, which are getting into the circulation and going elsewhere. So this is focusing the immune response onto this area. But remember, if it's out of control, that dendritic cell, which has picked up some sign of infection, is going to go to the lymph node and inform the adaptive response. And we're going to talk about that next time. Now, inflammation is good. When you have flu-like symptoms, it means you're having a good immune response. This is typically caused by viruses that kill cells. Cytopathic viruses we say activate the innate response because they kill cells. The cell pieces are floating around. They can be picked up by dendritic cells and macrophages. It activates them, and that makes for a good innate response. So cytopathic viruses have to have antagonists of the innate response, and that includes these viruses shown at the bottom here and many more as well. Some viruses do not stimulate inflammation. They come in you, they infect, but they don't cause cell damage. So there's no way, they don't cause cell damage, they don't cause apoptosis, so there's no way to sense that they're present. The dendritic cells do not pick up packets of apoptotic protein, and so they don't make a good innate response. These viruses also don't make a good adaptive response, and they typically make persistent infections. So these non-cytopathic viruses, they don't kill cells, they don't give you a good innate response, you don't get good inflammation, and you get a weak adaptive response. They can cause persistent infection. So all the herpes viruses that we'll talk about typically cause persistent lifelong infections. They're not cytopathic, they don't induce a good clearing innate response. So this is something we're going to revisit more next time and beyond when we talk about persistent infections. So the inflammatory response, again, heat, swelling, redness, and pain, means that the innate and adaptive arms are communicating well. And if you don't have inflammation, then you're not going to have a good immune response. And this is one reason why we put adjuvants into some vaccines. Some vaccines, especially the ones that don't replicate in us, that are inactivated vaccines, they don't kill cells because they're not replicating. So they're not going to make a good immune response. So we mix chemicals in them that stimulate inflammation, that give us those 4 inflammatory signs, and that help the innate response communicate better with the adaptive response. So that's just one reason why we use adjuvants. So let's summarize all of this. We have a mucosal monolayer. Virus has infected it. Those cells are producing cytokines. Some of those are interferons to try and quench replication. Some of those bring in dendritic cells and macrophages and other cells to the area to try and clear the infection. But maybe the infection keeps going. The dendritic cells have meanwhile picked up packets of protein from the infected cell. They're going into the lymph node where they're then going to ask T cells if in fact these antigens are foreign. And if they are, we're going to have antibodies and T cells produced. And that is actually what we're going to talk about next time. But let me end with this one phrase which you cannot forget. All viruses encode at least one regulator of everything we have talked about today, all the intrinsic and innate defenses. And you'll see next time they also encode antagonists of adaptive defenses.